My hope is to be able to both live up to kind of the hype from the number of times LD square regression has already been mentioned this week, and also provide enough technical insight to like the limitations and kind of the assumptions that are built in to have more awareness of what's going on. This is actually a more technical talk on LD square regression than I normally get to give, so I'm excited to dig into those details. So LD square regression primarily lives in this series of 3 papers in Nature Genetics, published all across 2015, and his work kind of co-led by Brendan Bulick-Sullivan and Hilary Finucane, with a number of people at the Broad, including my advisor Ben Neal and Alcus Price. And so kind of for the 2 lecture sections today on LD square regression, we're mostly just going to work through these 3 papers. And so the logical place to start is with this first paper on LD square regression itself. And it kind of focuses on this question. So Patrick showed you some of these QQ plots yesterday that we kind of use as a QC metric to think about population stratification. And kind of this challenge that both population stratification, where we have just kind of inflation of our GWAS test statistics, because we've haven't corrected for population structure. So the one of these that is population stratification, I'll let you guess which it is because we've done a GWAS where cases are all individuals from Sweden and controls are all individuals from the UK. And so we're just picking up all the random drift that exists between those 2 populations. But the other one of these is that we've simulated lots of small true effects across the genome. And we have true polygenicity, and if we tried to do genomic control or something to correct these statistics back to being on the 45-degree diagonal, then we're actually correcting out a bunch of true signal, and we don't want to do that either. So the question is, how can we tell the difference? And this becomes increasingly important as we get to our GWASs of really large sample sizes, so this is the example of the schizophrenia GWAS from 2014. Where as we continue to increase power for things that we presumably believe are actually polygenic, we're going to see increasing deviation from this line. And so identifying, even if most of it's polygenic, is there still some population structure or other confounding that's happening? What portion of this lift off this line is a problem versus how much of it is just that we're actually finely powered to detect things. So with that motivation in mind, we can kind of start building some intuition about LD and how we might be able to use that as a hinge to differentiate between polygenicity and stratification. We're going to walk through a cartoon example, and then we'll get to the math to kind of formalize these ideas. So let's imagine that this is our entire genome. There's only 20 SNPs. It's the entirety of our genetics. We've got each of our variants here marked along our single chromosomes genome. And we have LD blocks. So for cartoon purposes, we'll imagine these LD blocks are perfectly correlated. So these 9 SNPs, all perfectly correlated. You measure one, you know everything about all of them. And they're kind of interspersed. There's some lonely SNPs in between these blocks that aren't in LD with the other ones. We're simplifying for illustrative purposes, but the general idea that there are these blocks is roughly correct, and they are of varying sizes, and there are things in between them that don't correlate with much. So it's not too terrible of a cartoon. But so if we have this structure, then we can start thinking about what happens when we have a polygenic trait. Either in polygenic relative to the size of a genome of 20 variants or relative to a huge genome. And so let's say we have our polygenic trait, and we know nothing about the genome, and it's polygenic, so we've sprinkled the effects that are causal for that trait randomly. So this is like the true causal thing that if you flipped that variant, you would actually change the phenotype. And in this case, we've hit these 2 locations with that causal variant being related to our imaginary trait. Maybe it's preference for board games. And so if we then do the GWAS for this trait, we will see association for all of the variants in each of these respective LD blocks. So they're perfectly correlated. Anybody who has the change in this variant that is causal for whatever trait we're talking about will also have all the corresponding variants that are in LD, and all of them will be associated with the trait. So we'll see this pattern in terms of association relative to this pattern of causal SNPs. We can repeat this thought experiment for a second trait and throw down some more causal variance. This time we hit another pair of LD blocks. We can repeat it again for another trait. This time we hit one LD block, and we actually managed to hit one of the lonely SNPs. So we'd actually find the causal variant and not be stuck worrying about the difference between association and actual causal effects, and can find mapped perfectly to that locus. But the idea of this thought experiment is that if we continue thinking about kind of randomly throwing down effects on areas of LD and what association looks like, we can build through to this intuition that if you are a SNP that is in an LD block, you're more likely to be associated than if you're a lonely SNP. And in particular, it's proportional to how big your LD block is. So if you're in this LD block, 9 times out of 20, for our 20 possible places we can throw a causal SNP, you're going to be associated, assuming that you threw down the things at random. So this is kind of the key idea about how we're going to think about the relationship between LD and polygenic effects. So SNPs with more LD friends, SNPs that better tag their region. I'm told there was lots of debate over whether we could use LD friends as a technical term. But nonetheless, the more things you're in LD with, the more likely you are to tag a causal variant, just because you have more shots at tagging a causal variant, given that you imagine that causal things are distributed randomly. And so the more likely you're to be associated in a GWAS. So that's polygenicity. What about drift? Now the intuition here is a little harder to work through. But the idea is that if we assume our primary concern for confounding is population stratification, so these differences between populations that have had time to kind of breed more amongst themselves and with each other over long periods of time, and thus you get kind of this random walk of allele frequencies within each population, and at least in some cases, the random walk ends up in different places for the two populations. But if we then think about the mechanism for that random walk, if we assume that this LD structure exists, then those things are still correlated in those blocks. So unless recombination comes in and breaks up that LD, sufficient that we now have different LD structures in the two populations, then the things that are in LD will have to have moved together. So as a given variant, instead of the likelihood that you've drifted being related to your LD, the likelihood that you've drifted is equal because you will have drifted either with your haplotype block or not with your haplotype block. And the likelihood of that block drifting is not related to the size of the block. This is admittedly a little fuzzier in terms of describing what this is. It can be formalized under more special cases of population structure, but we're at least going to try and live on this idea that under pure genetic drift, we don't expect a relationship between LD and the amount of difference in allele frequencies between populations. If we start worrying about selection or differences in LD structure between populations, then this model starts to break down. We'll talk about that in a more formal sense a little bit later. But at least as a first approximation, under worries about pure population drift and that you've just done a bad job of matching what population your samples are from, this gets us a decent amount of the way. And just to kind of show that I'm missing slides. All right. Okay, so now let's get to a more formal model for this. So we're going to assume the same additive model that we've talked about a number of times already this week, that you have your trait of interest and that there's some collection of your genotypes that in an additive sense can predict this phenotype. And we're going to talk about this in the context of having a standardized trait and a standardized genotypes, just for simplicity's sake. Though we'll note that importantly, if we're going to start making assumptions about having a variance for these effect sizes, that will implicitly assume that there's a relationship because we've standardized between those effect sizes and the minor allele frequency. Given that when we standardize the genotypes, we're effectively doing an operation with minor allele frequency because of the 2PQ binomial probability. We can choose alternative forms for that assumption by sneaking in additional terms here for allele frequency. in practice, it's kind of a fuzzy question as far as where exactly you want to put that. We're going to hand wave that and say we're going to make some assumption about the relationship between effect sizes and allele frequency. Yes, Dan.
So just to clarify, the reason why this assumption is coming in is because we're standardizing the genotypes so that they all have variance 1 and mean 0. And then in addition to that, we're going to be assuming that the betas are drawn from the same distribution.
Yes.
Across all of the, all J.
Yeah. So if I actually flip to the next piece here, it's actually on the next slide. But yeah, so we're going to assume that we can say something about the variance of beta J not conditional on a specific J with a specific allele frequency. But kind of before we get to that, what we're going to say about that, the other piece to think about here is that so these are causal or at least pseudo-causal effect sizes. So if this is the sum over literally all your genetic variants, and there's no covariance between these variants in the population and other confounders in the population, then these are causal betas. Otherwise, they're kind of the best linear predictor in the population based on whichever set of variants you're putting in. So we're going to get to talking about this is the idea of heritability H-squared SNP. So heritability specific to SNPs is best variance explained by the SNPs, kind of separate from their actual causal effect sizes. But so the idea is that given these causal or at least tagging of causal effects, in the GWAS we're estimating marginally 1 SNP at a time. So we get, in terms of the effects, you get kind of through the hinge of omitted variable bias, you get a sum over the effects of all the SNPs you're correlated with proportional to how correlated you are with that SNP. So for SNP J, we get, when k equals j, obviously the correlation is 1, so you get its true beta from this form, but you also get the proportion of the beta proportional to how correlated you are. So this is our formalization of the idea that when you look at the association in the GWAS, you're getting this additional piece. And so the other component here is we're also going to say that it's possible that our betas are biased by some form of confounding. So mostly worried about population stratification, but we can also imagine confounding due to inclusion, family relatedness, or something along those lines. Yes?
So, let me know if you're going to get into it, but I wonder if any thoughts on the D-prime statistic of linkage and how it's not really directly related to R. And so, I mean, I know why you're using it for honestly, it makes more sense because the variance explained, but are you going to address the D-prime at all, or do you just not like it?
I don't have any problem with it. It's not the metric we want here for statistical reasons. So yeah, so there's this D-prime statistic that instead of looking at like a raw correlation in the classic sense, like Pearson's correlation between two variants, it's instead a metric of how often the variants co-occur on the same haplotype rather on different haplotypes. That kind of goes back to a more pure genetic understanding of linkage. So oftentimes if you're looking at software to estimate degree of LD, it'll give you the choice of R versus this D-prime statistic. In practice, for most of the statistical questions that we're worried about, we care about the correlation rather than the D-prime.
If you remember, so we actually derived that equation yesterday, just by the properties of OLS. So assuming we got the betas from the OLS, you can actually work this out in just like 2 or 3 steps, and it shows you.
Straightforward. It's the D-prime that's more complicated. That's why.
Well, yeah. So that's what I'm saying. But the R is what shows up in the standard OLS derivation of where this bias comes from, and so that's why it's the right thing in this particular case.
So here the worry would be partly from standardizing the variance to 1, but there are a variety of reasons why the distribution of the beta could be different depending on the frequency, and so I guess the question is, how much more complicated would the software be if you kind of assumed instead of, you still assumed normality and everything, but sort of a normality of the beta as a function of, with parameters that are a
built
function
into the software,
of the
I think,
frequency.
right? It's not deleted that with the alpha parameter.
Yeah.
Although it's not
like,
estimated, you fix it.
Right. So I'm trying to think how deep I want to dive into that well. So you can imagine.
That's in the software already, basically.
Yeah. So you can imagine that in standardizing the beta, you're doing something like you're doing a function of this, where p is your allele frequency, since this is the binomial variance. So you can imagine that instead of normalizing by this, you can normalize by some modified thing of this. So kind of scale alpha. And so kind of at one extreme of the logical values here is that you normalize so that you do the standard normalization, all of the x's are variance 1, and kind of once we assume a constant variance for beta, the assumption you're making is that the variance explained by each SNP is the same. So kind of implicitly then, if you're a rarer SNP, you have a larger effect size to compensate. The kind of other end of the distribution is you say you don't standardize at all, and that then you still have the distribution on beta. So as you you have a constant distribution with respect to effect sizes of an allele, and it doesn't matter whether that's a rare allele or a common allele, we're still going to say the distribution's the same. Those are kind of the poles which you can slide between by adjusting this alpha. There have been efforts in various places to estimate what alpha looks like in a range of traits. It's probably somewhere roughly in between the two ends, as you might expect. So there are indeed larger effects for rarer things, likely because selective pressures press larger effects down, at least for deleterious things. But actually nailing down what that value is, and it's going to be trait specific as well. So there is the option in the LD score software to make any assumption you want in terms of this alpha parameterization, in terms of what assumption you want to make when computing LD scores. But in practice, we tend to live on this end where we assume equal effect sizes, and then I'll talk about this in more detail later, but restrict what set of variants we look at so that we're in a domain of allele frequency where the differences between those models is going to be less consequential. All right. So we've got our causal effects. We have the betas that we've estimated in GWAS. Now, let's go ahead and make some assumptions about the terms in these betas for the GWAS. So the first thing we're going to do is we're going to say that these causal betas are random, and thus we can talk about the distribution of these causal beta sub k's. And we're not going to make a distributional assumption, at least not yet until at the margins, but we're going to talk primarily about moments. So we're going to say mean 0, variance of this h square SNP over m, where m is the j number of SNPs that you're summing over. We're going to assume that these betas are IID and that they're not correlated between SNPs. So the SNPs themselves can be correlated, but the causal effect of a SNP does not tell you anything about the causal effect of that SNP's neighbor or any other SNP elsewhere in the genome. Debate that. Well, it's a fairly strong assumption, but it's one that is necessary to make the method work through. We're also going to assume that the distribution of these effect sizes is not at all related to LD. So this is the intuition I tried to convince you of up front with the relationship on drift. The degree of LD tagging is kind of a random function of drift and not related to the effect sizes. Once you have selection, this becomes an untenable assumption, but we're going to hope that at least for common variance, the force of selection is weak enough that this is at least approximately right. And then we're going to make a couple of quick assumptions about our confounding, which is to say that it's also mean 0 and just define that it has a variance, and that it's not correlated with either our effect sizes or our error terms. So that our degree of confounding is not a function of the actual effect size. We're not seeing the things with effects be more affected by population stratification than random other things. And then it's not, we're not seeing more confounding for things that we're not able to measure as well. And kind of by OLS, we can make statements about the distribution of our error term being normal with variance proportional to 1 over n, where we're going to make this simplifying assumption that assuming any individual SNP's effect size is small, the residual variance should be very close to the full variance of y, which we fixed to 1 by standardizing our trait. So we're just going to simplify this to say that the variance of our estimation error is 1 over n. All right. So having made all of these assumptions, some of which we'll come back and try and relax in a little bit, but at least sticking to these assumptions is the first approximation. Let's look at what we have at our GWAS. Yes. Sure.
No relationship between the effect sizes for the different SNPs.
Mm-hmm.
So if I've got two SNPs that, so if I'm looking at something like, say, schizophrenia, I've got two SNPs that are in genes that affect something in the brain.
Yep.
I would probably expect those effect sizes would be related just because they're both dealing with a similar pathway, even outside the LD structure between them.
Yep.
But we're basically assuming that that's not the case, right? So if I've got a SNP that's associated with, so my prior is that if I've got a SNP that's associated with a gene dealing with like nails on my feet or things in my brain, my prior is that the effect sizes of the two are not related except through the LD structure between the two. Is that right?
Right. And so there's kind of a couple things we can say about this. One is that given that we're talking about this in the context of the sum of things genome-wide, one way to try and hand wave our way into saying we're comfortable making this assumption is just to say that you may indeed have information about your closest proximal effect sizes, but at the margins of thinking about all effects, there's not a ton of correlation to worry about. That's still a somewhat difficult assumption to make because you're mostly worried about your correlation with things that you're in high LD with. The other solution is when we get to the partitioning heritability, we're going to directly weaken this assumption in that it'll be that there's no correlation to the effect sizes conditional on some outside information about the SNP. So conditional on, saying that they're in a coding region that's something brain expressed, then there's no correlation.
I think there's also another thing that helps us a little bit. In that so the concern is that you have maybe a pair of SNPs and they're on the same gene or something. So we think that mutations on that gene are both going to affect the outcome. So that's problematic. But because we're arbitrarily selecting the reference allele, it might be that you have a large effect for one and a large effect for the other. But it could be that they go in opposite directions. So if the relationship between whether or not the sign of the effect is the same is independent of the sign of the LD between the SNPs, and I think you can end up with this, this as well.
So couldn't you test this by just running this?
Yeah. So they've done work on this. In general, this assumption doesn't seem to hold very well, I think, but it also doesn't seem to break the estimates from LD score regression horribly either. But you should understand that that's the most important assumption of LD score regression. It probably doesn't hold, but the benefits of LD score regression that we're going to get are so strong that we're willing to just accept that it tends to be a little bit better. So if you just want to get heritability estimates out of LD score regression, then it's complicated. Maybe this is breaking things. But as Raymond's going to talk about, if you want to do things like get genetic correlation or partition the heritability, some of these biases cancel each other out, so you might be more happy with them.
Yeah, I think that's exactly right. There's a reason that we're kind of glossing over the heritability estimate that we're going to get out of this and focus primarily on partitioning and correlation. And it's partially because these assumptions are fairly strong, and thus, thinking about how that affects your actual estimate of the heritability, the SNP heritability is kind of rough. All right, so kind of the thing to note here is given that our quantity of interest, at least in theory, is the SNP heritability that lives in the variance of beta, it might be of interest to think about things that are squared in the betas, which leads us naturally to the chi-square statistic. So from GWAS, we have our chi-squared. That's the square of these betas over their sampling variance. And given the statement that we made on the previous slide, that the sampling variance is very close to 1 as long as we're talking small polygenic effects, then we just have 1 over n for that, and thus our chi-square is approximately equal to n times the square of the beta we just defined on the previous slide. All right. So now let's take some expectations for this chi-square. And by virtue of all the assumptions we've made on the previous slide to try and get rid of the cross term, so saying that the confounding is not related to effect sizes or the error, that each of the betas is not related to the other betas, we can kind of ditch all the cross terms and get down to just expectations about the squares in each of these terms. Having done that, again, because there's no relationship between these betas and the degree of LD, and that the betas are mean 0 in expectation, we can extract this to be just our single variance of beta. We've previously defined that we're just going to call the variance of this S term A, and that the variance of our error term is approximately 1. And then the last thing we're going to do is kind of plug in what we said on the previous slide, that the variance of this beta j is equal to this h square over number of SNPs. Cool. So what we're going to do is, we're left with this function that's related to the sum of the r squares. And we're going to say this sum of r squares, so for SNP, this is for SNP j, the sum of your correlations to all the other SNPs. We're going to define this as the LD score of a SNP. So it's the sum of your correlations, this idea that we started with in the intuition, your number of LD friends, but we're being a little bit nuanced and not just counting LD friends. We're actually measuring how much LD you have in total to things. And we're going to call this the LD score so that we can plug this in to the equation we had on the previous slide and end up here, which I've heard other people call the fundamental theorem of LD score. I'm not sure I'm comfortable with putting a pedestal like that, but this is indeed kind of the core equation that we live around in LD score, and we kind of build off of this in various directions. And so we can show that this works. We can look at, do we see expectations that scale with our defined LD score, depending on whether or not we have polygenic effects that should scale with the LD score, or if we have stratification that should be constant regardless of your degree of LD. So here's the two QQ plots I showed you at the beginning. So if we take this top one, and now what we're plotting is your LD score along the x-axis. We've taken bins so that you can look at averages within each bin, and then your mean chi-square for SNPs in that bin of LD. And so in this case, we see there's no relationship between that LD and your mean chi-square, and indeed this is the data that was simulated with population stratification. Down here, we do the same thing, and in this case, we have this nice linear relationship between the degree of LD and the mean chi-square, and indeed, this is the simulation that has polygenic effects. So at least as proof of concept, under the model that we specified, we do get this nice linear relationship. So now we can take our kind of cryptic level of stratification and start looking at whether or not this is consistent with population stratification or consistent with polygenic effects. Cool. And so kind of the way to drop our estimates of these things out of here is that from our GWAS, we have our chi-squares, and we can compute LD scores for each of our SNPs, and presumably we know the number of SNPs and we know our sample size.
Sorry, and the LD score you're getting from a reference sample?
Yeah, we'll get to that in more detail in a minute. And so if we just regress our chi-squares in the GWAS on this function of sample size and our LD score, then we have our intercept, which gives us our estimate of this term here, this 1 plus this function of confounding. And so this is the quantity that Patrick and I talked about yesterday that you can use to correct for just the stratification. Similar to what you do with the genomic control and inflating your standard errors. So to the extent that this is greater than 1, it's a sign that this alpha exists. You have some confounding that has some variance and can adjust for that. And then the slope of this regression, in terms of how much it tracks with the degree of LD, gives us at least a first approximation of an estimate of heritability from SNPs. There's a nuance here that this is on the observed scale. So if you're working with dichotomous traits, there's some additional math you have to do to convert this to something more analogous to a liability scale, heritability, particularly if you're dealing with oversampled cases. But as long as we live in continuous traits, this is fine as a heritability estimate. And given that we're regressing a set of things that are correlated, because we're plugging in all of our GWAS summary statistics so that LD still persists for the association between SNPs, the standard OLS standard errors aren't going to work. And so we instead do standard errors via block jackknife. So we kind of chunk up these local regions, do a jackknife, and use that to get our approximated standard errors. It's probably not perfect because you're then capturing randomness that exists between sets of SNPs rather than the randomness that exists between resampling individuals. But given that we're working from summary statistics, there's not really a clear option on how you get the randomness that exists from resampling individuals. And so we use this at least as our approximation.
So if you add the PCs, that intercept term goes way down?
Yes. In general, you can, well, actually, I should, how do I want to quantify that? So in the general case, yes, if you have, if you start with the GWAS with no PCs and then add PCs, you should see this drop substantially. In a well-controlled GWAS, most of the time, you're close to 1, not quite all the way down to 1. I think we see in most cases. I think you guys have, at least I think in EA it actually has managed to get down to virtually identically to 1. But 1.01, 1.02 are probably the mean across published GWASs. But there's kind of cryptic things that if you start adding PCs that are noise, or if you have PCs that are not well tagging the actual effects, at some point you can actually be introducing reverse confounding. So we have seen cases where reducing the number of PCs actually improves the intercept for reasons that are kind of cryptic. Yeah.
Just on the slide 2 previous, I think, I just wanted to ask, the thing you said about...
back here.
The thing you said about the H, there being a square in the H squared, and so we look at the chi-square statistic instead of the Z statistic. I didn't quite follow that, because isn't beta, isn't H squared, isn't it already squared, as in, sorry, maybe it's a slide before that.
I think it was back here where we...
Yeah, back here.
This transition as far as why I went from talking about these GWAS betas to our chi-square statistics.
Yeah.
So to some extent, yes. There's a bit of weirdness that we talk about H square because it's a variance component, so we put a square on it because it makes sense to put squares on variance components. Maybe. But in practice, there's not much interpretability of H as a value. I can't say I've seen any literature where anybody's interested in the square root of heritability. The other practical reason is that if we focus on the betas, then we actually worry about having results that were generated with a standardized phenotype and a standardized genotype to have the betas on the scale that we're talking about here. Whereas if we move to chi-square, if the GWAS was done with some unstandardized or some partial standardization or whatever, all that will live in additional terms here that track that variance and kind of cancel out so that it's equivalent to if we had done the standardization. So by working from chi-square statistics, we don't have to worry about the actual measurement scale used in the GWAS and can just talk about things on this idealized scale of betas that track directly with the total variance.
Can you say just a little bit more about the standard errors and the block jackknife?
Yeah. In terms of how they're generated or why we might be concerned about them or what? Okay, so yeah. So in terms of how they're generated, so for those who aren't familiar, jackknife is kind of a special case of a bootstrap, where instead of dividing things, so first we're going to do a block version. So we're going to divide the genome-wide results into blocks that are kind of regional and should be at a size that they capture the local correlation. So one thing we could do is we could bootstrap. We could take, random resamples of a number of chunks of the genome, re-estimate heritability, and do kind of the standard bootstrap procedure. Jackknife, instead of doing that kind of randomly, we more systematically go through and estimate dropping one block at a time. So you drop, the first megabase of the genome, and you estimate heritability for the rest of it. You drop the second megabase of the genome and estimate heritability from the first megabase plus the remainder, et cetera. And there's nice analytic formulas for how this gets you to get you back to the proper thing that you would get with the bootstrap. But since you've done something systematic, you now have a more systematic standard error. It's not random in kind of the luck of the draw of your bootstrap. So plus it's a little more computationally nice. So that, in terms of the mechanics of what we're doing, that's what we're doing is kind of resampling. But as I said, since you're doing chunks of the genome and resampling in that way, you're in a sense capturing the degree of randomness across SNPs. So for polygenic things, this is at least kind of useful because you expect that there should be effects everywhere, and so you're dropping out some random chunk and re-estimating, and you're just trying to get the slope of this relationship with LD, and so the amount of randomness that's kind of consistent with there being some pseudo-randomness as far as what SNPs made it into your GWAS is captured, but you're not, as I said, capturing the kind of thing that you would do if you were doing a more classic bootstrap where you resample individuals and rerun an experiment. So you'd resample, number of people in the GWAS, rerun the GWAS, then redo the heritability estimate. You're not capturing that piece of the randomness.
So I'm concerned about chi-squared J's being correlated. That's because of the LD structure and us looking at SNPs that are in LD, or is that because of residual concerns over kind of correlation arising from our assumptions being violated in the model?
So this is primarily about the SNPs themselves being correlated due to LD, and thus there's correlation in their association results. So these individual chi-squares we're plugging in to do our regression are not independent of each other, and so our standard treatment of that for OLS standard errors doesn't work.
So because of the LD structure?
Yeah, because of the LD structure. So yes, you could conceivably instead do LD score regression with some set of pruned SNPs that are roughly independent, and then at least think about doing the OLS standard errors. You lose power because of the greatly reduced number of SNPs. You're not tagging as many things because remember this is all hinges around the degree of things you're correlated with. So to the extent that you're tagging things basically you're going to have to extrapolate from whatever set of things you put into the regression to the set of things they're correlated with. So that can be a bit tricky, but at least in principle that would get around to this particular question in terms of the LD structure.
Sorry, can I ask one?
Yeah.
So if I do this procedure, if I correct my standard errors, but I do it two ways. So I do it first with the breach SNP, with the SNPs that are in the LD, in LD with that SNP, and then I exclude those and do it with only the SNPs that are not in LD with that one. Would I see, I don't know if you've done this, but would I see the standard errors change very much when I exclude the SNPs that are not in LD with those?
This is not an experiment we've done.
Should be close,
You have an
right?
intuition?
It's gonna be,
I'd expect it to be
Because
close.
the assumption is about no correlation.
yeah, so I mean, I guess one implicit assumption that you're making when you're doing the block jackknife is there's not correlation across blocks,
That's right.
and that's not quite true. Like there's going to be SNPs on the boundary that are correlated. But if your blocks are big enough, then that's not going to be, that shouldn't be too big of a problem. In general, we do like 100 blocks, or how many do we do? Like we're doing pretty large blocks.
I think it's 200 blocks for the genome is
Yeah,
the current
and so
default.
these blocks that we're using are much larger than what we think the LD blocks are, and so this fuzziness of the boundaries isn't going to hurt us too much, I don't think.
I guess I'm wondering about whether or not the standard error correction is really capturing the correlation between the SNPs and LD, or is it actually dealing with this residual correlation because the model is misspecified and assuming that SNPs are uncorrelated except for LD.
I mean, it's possible it picks that up. It's just not something we've evaluated in that framing. I think your hand is next.
How much of this depends on the degree of polygenicity? I mean, have you I'm guessing different traits would have different degrees of polygenicity.
Yeah, so this is highly dependent on degree of polygenicity. So we talked about some in terms of what that means in terms of the assumptions, though, since we're assuming moments, at least some of in terms of expectations, it still works. The main place we tend to see the polygenicity assumption pop up is that you can start at the extreme end where you've got something that's monogenic, there's only one locus. And so rather than having this kind of random beta draw where you have enough room to get the expectations to start working across, many things, really all you have is the signal at that locus, and so the LD at that locus is the LD at that locus. And because you haven't actually drawn many random things, it's kind of a crapshoot as far as how much LD there is at that locus. And so what estimate you get is kind of all over the place.
But it's more about precision and not about I think an expectation you'd still get the right thing.
Sure. Well, I mean, in the expectation it's still right, but the quality of any specific estimate is questionable. That tends to show up nicely in the standard errors, though, in that since we're jackknifing sets of SNPs, you'll very clearly see that in the jackknife that drops your one significant locus, or if it's a few scattered things and it's just not highly polygenic, in any of the jackknifes that drop the high effect regions, you'll have a huge change in the amount of heritability that's left, and thus you'll get fairly wide standard errors that note kind of that lack of polygenicity.
But again, that just goes back to precision and not
Yeah.
bias. And so there are architectures that LD score regression just won't be as powerful to be able to deal with, but it's not inappropriate because we've not made any assumptions about the actual distribution of the effect sizes, just about the moments. And so we should be okay in theory.
Exactly. Are there other hands? Yeah.
Patrick, can I ask you a question? I mean, or Raymond.
Sure. Totally fine.
Because, I mean, it goes back to Patrick's comment about how blocks are not fixed, and then you can, be playing with the width of the blocks. So is it possible that because you can be making the blocks as small as you want or as big as you want, you can basically eliminate all the SNPs that are going to be correlated with the lead SNP or the SNPs that you're interested in, and that's why there would be no correlation between different betas. Is it something feasible, or?
So in practice, we don't really play with the number of blocks. We just
So it's fixed.
Yeah. So I mean,
Yeah.
you could do it, but the LD score software, can you even tell it to do different size blocks? I don't even know if that's an option.
I think you can change the number of blocks,
Oh,
but not the
okay.
size, not directly pick the size of the blocks.
If I saw someone, if I read a paper, and that paper said that they changed the option about the block size that they did their standard errors, I would become very suspicious. Because hopefully it would be robust to whatever you pick, and if it's going to be robust to whatever you pick, then the default settings should be fine. So if you're going to tinker with that parameter, you need to have a really strong justification for it. Otherwise, I'm going to think that you just kind of picked whichever one gave you the best result.
This theory about this for delete D jackknife in general, that there's certain values that would be too when there's like correlation between nearby observations, there could be a number of SNPs or whatever that you delete that could be too small, but once you cross a certain threshold, it should be fairly sensitive to it.
Who developed this LD score technique? Was it like biochemists or?
There's people in the Neil lab.
Yeah,
And
so
the-
statistical geneticists and statisticians, give or take.
And so they were mostly they were developing this in the first instance for like disease phenotypes.
Yeah, so like I showed you the schizophrenia result in part because, results like that are where a lot of this conversation was
But still complex traits.
oriented.
So not like disease, but not Mendelian diseases.
Yeah. So we have had people attempt to do this in like specific LDL measures and like specific metabolites, and it's kind of questionable how well it actually works. But as Patrick said, that in expectation it works. It's just the standard errors are often, you expect that they're going to be large enough that how much information you're actually getting out of doing this is questionable. It still probably works for getting an intercept though.
I think Brendan recommends either 100 cm or 1,000 cm for the block size. What's the standard that?
Yes, so there's the block size here, which I think is coded in terms of just number of blocks, and so it's kind of the actual size and genetic distance varies. Then there's the size in which you compute LD, which the recommendation is 1 megabase. I think that's actually my next slide. So yes, I talked about these LD scores as if they exist and are available. There's a lot of kind of things that come up in actually computing them. The first is that it's important that we have the unbiased estimator of our square. So if we take just the correlation constant squared, there's actually an upward bias. This is a standard issue with why you have adjusted R squares. And so we want something that's unbiased because we're estimating the degree of LD from some finite sample, and so that matters. Not too terrible. But given that we're saying that we're working from summary statistics, we don't actually have anybody's raw genetic data to compute LDN. So instead, we're going to estimate this LD based on some reference sample. And so most commonly, this is the European ancestry individuals out of 1,000 genomes. A nice public data set that works as far as reference for population structure. And so we're having to make the assumption that the LD structure that exists in these individuals is a good proxy for the LD of the individuals in our GWAS sample. So in practice, that means we are now restricted to running this analysis on, say, a GWAS of European ancestry individuals. There's some claims that you can, there's some people who will claim that you can get away with it on kind of add mixed samples. It's kind of mixed. Depends on how much you want to actually interpret the heritability estimate. But nevertheless, you are kind of attached to a given population with this. Trying to look cross populations is not recommended. But being able to do this out of a reference sample is kind of the value of LD score regression, given that there are other ways we could estimate heritability if we actually have the raw genetic data. So we can do the Gremmel method that's in GCTA. We can do Hazeman-Elston regression. I think at least Gremmel is getting covered sometime next week, I've been told. I think Matt Keller is going to talk about it. So you'll hear a little bit more about some of the other alternatives if you're actually interested in the H-square SNP estimate. But for LD score purposes, we're going to focus primarily on the downstream applications here and on the ability to do a summary statistics, which is why we're doing this. I'm going to go with the question back there first.
So I'm guessing even with the 1,000 genes, there will be some degree of variation in the LD structure, subpopulations
Yep.
within the 1,000 genomes. And if your GWAS is based on some of these subpopulations, which there are some sort of basic small differences in LD structure, would that also not subsequently influence your intercept? I mean, if it's not an ideal match between the LD structure of the population in your GWAS and the LD reference LD structure.
So there's not an expectation that it should influence your intercept. So if we're wrong about the LD structure, or at least we're off by a bit, what we're talking about is estimation error in these L sub j's, basically. So as long as we can approximate that as being random error, then essentially we have a weaker predictor here. We'll have attenuation of our estimate of the h square SNP. But that shouldn't affect where the intercept is. If there's directional biases, then we're in a little more trouble, which can exist if you're talking about populations with different, like, average levels of LD, which do exist. I see you want to make a comment.
I think the intercept, I mean, like, the slope attenuates, but I think that the intercept tends to also be inflated when you have noise in your covariate. I think that's in general what yeah. I mean, think about, like, the infinite case. You have infinite noise, then the whole curve is going to rotate about the mean LD score, right?
Yeah, it's fair. It does actually. The estimates are
the intercept
correlated, so you will
be
still see
inflated
some.
if you're using the wrong reference.
Yeah.
sample.
In practice, we haven't seen it occurring, at least to a level that matters.
This LD pred for all the betas as well. So if you get the wrong LDs, you're going to be messed up on the
Well, so this is
on
different than
betas
LD
and not
pred.
just the
Yeah. So we're using GWAS betas here. So LD pred we'll talk about next week for prediction.
Yeah, that's another important consideration here. Is we're assuming raw GWAS betas. So if you've done LD pred, if you've run a mixed model, if you've run some other method that is not a GWAS, all the math changes, and in ways that we haven't fully worked out. So important to keep that in mind as far as input. In terms of more like specific ancestry matches for LD, certainly you could start looking at the subsets of different populations from Europe that are available in 1,000 genomes, or looking for other reference data. You're going to pay some price in terms of sample size, in terms of precision. So, I mean, the European ancestry out of 1,000 genomes is already only a few hundred individuals. There are larger reference panels available / on the horizon, depending on what data you have access to, that should make the situation much better for not only European subpopulations, but also kind of more global populations. But this is standard enough at the moment that we live on top of it, and it seems to behave mostly okay. All right. The other point here is that it's very important what set of things we're summing over in the sum over j SNPs. So when we compute this L sub j in whatever reference data, we can choose to sum over any of the SNPs that are available in the reference data and generate a score for any SNP that's available in the reference data. When we then go to do our LD score regression, we're going to run the regression with whatever set of SNPs we've computed an LD score for and is also present in the GWAS. And so the kind of key distinction here is in terms of the universe of things that we're estimating for. So the kind of theoretical case is that the h square SNP we're talking about has been derived based on being the h square by all the things that we sum over when we define this L sub j. So we can define this for whatever choice of set of SNPs in the reference population that's not necessarily present in the GWAS, which means that in terms of how we're then computing, in particular, this will hinge off of, if I flip back a slide, this will hinge off of what statement we make about M, which is nominally the number of SNPs we've summed over. So whatever SNPs are in the regression, we're assuming that their results extrapolate to the remaining SNPs that we used in computing LD scores but weren't necessarily in the regression. And this, where this is important primarily goes back to this point about allele frequency. So in particular, something like 1,000 genomes or other sequence data has a lot more density of rarer variants. So you could end up in the position where if you just include like every variant that's present in 1,000 genomes and do this sum here, you're then assuming that the effect sizes from your common variant GWAS extrapolate to the full spectrum of rare variation that you included in your sum. And because that's a very strong assumption, generally we will choose to restrict what we sum over here to common SNPs so MOF greater than 5%. As well as in terms of the regression, well, you can either exclude in the regression or you can exclude here. Excluding the handful of regions of the human genome that are characterized by kind of absurdly outlier densities of LD. So this is in particular the MHC region on chromosome 6, the chromosome 8 inversion region, and there's a longer list, though we tend not to exclude those. And the reason is just that if you include these, you end up with outlier values of L sub j for the sets of SNPs in those regions, and we know that OLS performs poorly when you have outlying values, so we don't want to do that. The other piece here is, since we're extrapolating from GWAS SNPs, we're also assuming that GWAS SNPs are representative, which if you've run a GWAS with some targeted chip, so something like exome chip, metabo chip, immunochip, psych chip, though it at least includes a GWAS backbone, something that it targets regions that have higher concentrations of effects, it's probably a bad idea to extrapolate from the results you have for those sets of SNPs where you already expected high degrees of effects and saying that the rest of the genome has that. And so generally, you only want to do this where you have full genome-wide data from some kind of generic GWAS chip rather than some targeted chip. Yes?
Can't you talk about the psych chip, you say, but also a backbone of GWAS? So psych chip, what does the psych chip have? Just like variants or something?
Yeah, so the psych chip is, I don't have the numbers perfectly committed to memory, but it's 250K variants of a generic GWAS chip. I think 50 or 100,000 variants worth of exome chip. So that's higher density coverage of gene coding regions. And then like 50,000 SNPs targeted in various ways to psych-relevant content. So this is a mix of regions that had shown association with various psychiatric disorders in the earlier PGC studies, some genes that were considered candidate genes based on sequencing studies and other previous candidate gene work, and a few instances of some really dense coverage of genes looking for loss of functions in those genes that were predicted as potentially occurring, but had never actually been observed in the population. So it was, they called it was kind of experimental content for sequencing by genotyping. In practice, we're not seeing much off of it. And if you restrict to the common content on psych chip and do standard imputation and stuff, it behaves mostly like a generic GWAS chip. So we do, in practice, run LD score stuff off of results that include psych chip data. But yeah. Yeah, it's somewhere in the 400, 500K range. I forget I want to say it's 500K, but I forget the exact numbers.
I think the 250 generic GWAS is that enough for, let's say, population stratification?
Yes. Well, so for stratification correction, we can actually get away with somewhere on the order of 50,000 variants, which is what we end up with after, say, LD pruning off of what's genotyped on psych chip or most common chips. The 250K is kind of the lower bound of what gets called a standard GWAS backbone. So, to be able to tag all the common variation in the genome in at least reference samples, which primarily means Europeans, to some degree of capturing most of the common variation. That's kind of what's been worked out as the lower bound that Illumina or Affymetrix or whoever will sell you. 500K is definitely better. There are chips that have 1 million or more SNPs that will just continue to increase density and coverage of additional populations. But yeah, in terms of thinking about GWAS chips, generally you're talking about 250,000 lower bound.
And is there any benefit to getting SNPs that have been selected for being informative for ancestry species? Do they limit that?
Yeah, so there are chips now that target having more coverage of ancestry. There's not much of that on psych chips. There's not much of that on the older chips, but there are chips that are targeting more coverage now for ancestry purposes, both better capture ancestry and to just to get better information for studies in those populations.
So could you say a few words about Gremlin, Husman, Elston? What are they?
how to boil this down to a few words. So Gremel lives off of doing an analysis similar to a mixed model like Patrick described yesterday, where you're building this matrix that's kind of the low-level genetic similarity between individuals and looking at the extent to which that kind of low-level genetic similarity is reflected in trait similarity. And
It's like a twin study, but in like unrelated people. So you're just saying
Yeah,
people
exactly.
that seem to appear to be the same genetically, then do they appear to be the same phenotypically? And if that's a strong relationship, then you say it's highly heritable.
Yeah. And then Hazeman-Nelson is similar, but instead of fitting a variance components model, you're doing a regression of the elements of this genetic similarity against the elements of looking at phenotypic similarity. So all those pairwise cross products. You can work out special cases where Gremel, Hazeman-Nelson, and LD score are all asymptotically equivalent. There's a white paper from Brendan Bulick-Sullivan that's on bioRxiv, and I don't think is actually intended to be published anywhere other than bioRxiv at this point. But that works through kind of the math and all the things you have to think about in terms, largely in terms of the universe of SNPs each of these things are covering in terms of talking about H square SNP as a quantity. All right, I'm gonna try and get through a little bit more, and then we're probably up against wanting to take a break. Okay, other nuances in actually doing this. So thus far, we've made no distributional assumptions. We've only talked about moments. But we can imagine there's a couple things where weighting the regression would be useful. One is that we've talked about the expectation of chi-square scaling with LD score, but also the variance of the chi-square statistic is going to scale with the LD score. This was somewhat evident on the plots I showed you earlier and becomes more evident as you look at like not binned LD scores. Not binned chi-square statistics. So adjusting for that heteroscedasticity would presumably improve the efficiency of the estimates out of a regression. The other point is because there's this correlation between SNPs, you have this issue with the standard errors in that you have the points aren't independent in terms of getting standard errors. But also for areas of high LD, those regions are being more strongly represented in the regression. So in some ways, you're overcounting the information in those regions compared to the other regions. Kind of the thought experiment here is what would happen if you picked some section of the genome and added in a duplicate copy of every SNP that was like your pseudo SNP that was imperfect LD with the actual SNP. And now you'd have all that extra representation in the regression that's not actually new information, but still affects kind of the set of chi-squares that you're looking at. So to kind of adjust for these two things, we can do a weighted regression. And so for the heteroscedasticity, if we go ahead and now go back and make some statement about the distribution of the causal betas and say that they're normally distributed with the mean and variance that we already specified, ignore the confounding piece, assume that in most GWAS the confounding is well controlled, and say that n is large enough to get us to central limit theorem purposes, then you can show that the variance of the chi-square conditional on the LD score is this function here. So you in a sense have a scaled chi-square distribution. And so in practice, what this means is you can generate some rough initial estimate of the SNP heritability without weights, to plug that in and then have the requisite values to generate the weights for this variance and weight inverse to this variance in terms of doing your final regression. And we combine those weights with the weights for the correlated SNPs, which is done on a much more heuristic basis, which is to weight by the LD to other SNPs that are in the regression. So the concern about overcounting is that you're overcounting because you have multiple SNPs in the regression, which may or may not reflect the amount of LD to things that were in your reference sample. And so we do this by generating an LD score that's specific to SNPs that are part of HapMap 3 on the assumption that the HapMap 3 common variants are well represented and easily imputed in basically every study at this point. And so you can assume that this is a decent proxy for the amount of LD you have between SNPs that are present in the regression and can weight relative to this as well. Again, this is a very heuristic correction, but This is what seems to function at least decently well. All right. So I told you all that about univariate heritability, so we can get to partitioned heritability. I'm a little behind where I wanted to be in terms of breaking, but do we want to take a break now, or do we want to power through at least the initial talk, the initial ideas of partitioning before we take a break?
Why don't you just explain the basic idea and then we'll take a break.
Cool. So, second paper, taking this idea where we've done the work, we get our nice control for confounding in terms of this intercept term that we can use to evaluate stratification. But now let's talk about partitioning in terms of more useful things for this heritability. So we previously talked about having this constant heritability, this constant variance of SNPs. So essentially the heritability from each SNP is on average about the same genome-wide. And as kind of has already come up in conversation, this is a very strong assumption. It also kind of plays into this assumption about that, genes in the same region, variants in the same region that are associated with things that may have stronger effects would also then be correlated by virtue of being in these regions. So all sorts of places where there might be an issue. So talking about the coding variants versus introns or energetic variants, or the things we don't want to call junk DNA but do tend to have smaller effect sizes. We can think about this for relevant to a particular trait. Brain genes maybe were relevant than muscle genes, to the extent that we can actually call anything a brain gene or a muscle gene, but that's a digression. Also, we've talked about how we had to make strong assumptions about the kind of functional form of effect sizes with relationship to allele frequency. We might want to allow that to vary and estimate what that distribution looks like a little more empirically. And so essentially all this leads us to wanting to do this partitioning model that will allow us to allow for violations of this. So let's talk about there being some regions that have a higher variance of effects on average, and other regions have a lower variance of effects on average, and potentially find interesting things about what we can say about the regions that have higher variance of effect sizes. So if we go back to our cartoon, we've talked about we have our sum of the LD across things. So again, in our cartoon example, our LD blocks consist of variants that are all perfectly correlated. But so in our cartoon genome, we can now imagine that we know that there are some of these variants that are a DNA hypersensitivity site that lives here inside this LD block. We have some coding SNPs for the one gene in our fake genome that are over here that kind of spans a couple LD blocks plus this lonely SNP in the middle. And so that we can imagine, instead of computing our LD score as a sum across kind of all variants you're in LD with, you can limit to summing across variants that are in specific categories. So we could have our score of just your LD to these DHS sites. You could have your score of your LD to just coding variants. And this gives us kind of the beginnings of a way to think about splitting up the heritability that exists in each of these categories. And so mathematically, we're going to get at that by saying that instead of this flat amount of variance that is the same for all SNPs, you have the variance of the causal effects for a SNP is given by a sum over some variance term for each category. And so the easy case of this is if we have disjoint categories, so there's no overlap, and we assume that there's some category that's kind of the catch-all, everything that's not in the category we're interested in category, then your H-square SNP particular to a specific category is the variance of the effect sizes in that particular category, conditional on making all the same assumptions we made for the rest of the model in kind of the univariate case for LD score. And thus, your heritability here is this pseudo heritability per SNP in that category, this tau, times the number of SNPs that are in that category as part of this sum. Once we're into overlapping categories, then we are making an assumption that we can sum kind of the variance components. And so say that if you're somewhere that is both a coding site, you're encoding part of some gene, and it's an evolutionarily conserved region, and you want to say the conserved regions are also have stronger effects, that if you're in a site that's both of those things, then your heritability per SNP on average, this variance, is equal to the heritability per SNP on average for just coding SNPs plus the heritability on average for just conserved SNPs. So it's still a fairly strong model in terms of what you're saying, but at least allows more flexibility about these different categories and how there might be differences in distribution of effects based on annotations across the genome. And so we can kind of just quickly get through the math on this. So we go back to our same marginal GWAS betas, our same base model. Everything's the same up to here in terms of looking at expectations. We're making same set of assumptions other than having kind of now having conditional distributions of beta. And so what's changed now is that instead of this just being a single variance term to extract, we're now extracting to this set of taus that correspond to the kind of contribution of each category to the variance and effect sizes. And then instead of a single LD score, we now have an LD score for each category. So this is summing across this, the LD to the set of variants that are in the category. So as shown in the cartoon, you're kind of, instead of summing to your whole LD block, you're summing to the set of SNPs that you're in LD with that are part of some category that you've annotated. Right, and so kind of that leaves us to a question that looks very much like our quote-unquote fundamental theorem of LD score. I don't like it, but it's useful. And instead of a simple regression, we now have a multiple regression where we have our set of categories that we're regressing against, and each have their own LD score that's, again, computed from our reference data based on our choice of annotations.